Today's lecture is going to be talking about microscopic urine sediment, and this is going to be a procedure that you're going to perform in lab. And to save a little time there, we're going to talk about it now so that you have a general idea about what we are going to see when we perform this procedure. The actual steps that are going to be done we'll talk about in lab, but here we're going to get a lot of visual identification stuff. For the record, it might be helpful for you to have your book out in front of you because some of the pictures that I have might not be as clear to you. They have some very great pictures in there, so just open up to, I believe it's chapter 14, and kind of follow along. You can find these pictures flipping back and forth, and if you do have trouble, just let me know and I can guide you in the right direction. So our goals for this discussion here are to, number one, visually identify some clinically important stuff that you're going to see, potentially see, in a urine sediment. And that could be cells, it could be something known as casts, it could be crystals, or other artifacts, which we will talk about. And then after you've been able to identify them, we're going to have you try and link what they are to what might be going on in the patient. What is the clinical significance of those things when you see them in the urine? It's also really important to be able to tell what might be something normal in the urine and what might be abnormal, because it's totally normal to see some of these things and you don't want to panic if you do see them and report it out as something being wrong. Now, as you're going through this is just a very beginning preliminary discussion on these topics. So there's a couple different levels of understanding you can get from this. The first thing that I really hope, the base that I want you to come out with, is that you have a general idea of what the difference between the cells, casts, crystals, and artifacts are. So if you see something when you're doing your lab, you should be at least able to say, oh, that's a cell, oh, that's a cast, oh, that's a crystal, oh, that's something totally different. So that's the lowest level of understanding here. Hopefully, when we're done, you're going to be able to give a specific ID with a little bit of practice. So you're not just going to say that's a cell, you're going to say, oh, that's a white blood cell, that's a red blood cell. Well, that's a calcium oxalate crystal, not just a crystal. And then when you get really good, as I said before, what you're going to find is that you're going to be able to say, why am I seeing those calcium oxalate crystals? Well, potentially this patient has a kidney stone. So you can correlate what you see with disease. And that's really the highest level of understanding that we're going to get you to, just maybe not quite yet. So looking at the lab, one part, as I said, of a routine urinalysis is a microscopic sediment examination. And what you're looking for are, as we call them in the lab, formed elements. And those are the things that I mentioned before: cells, casts, crystals, artifacts, things that are particulate, things that you can see. You can't see dissolved urea in the urine, but you could see a red blood cell. So we're going to look for microscopic solid particles that you can visualize under a microscope. What will happen, in just the gist of it, is you're going to get a sample of urine, usually about 12 mils. You put it in one of these fancy tubes here that has a conical point at the end, and you're going to centrifuge it for 5 minutes. You're going to discard the supernatant at the top, and you're going to keep all the stuff at the bottom, which is the sediment. And you're going to kind of mix it up, and we're going to look at it under a microscope. It can either be stained or unstained, and that's how you're going to identify some of these things. So what we're looking for is this little part of the urine that gets concentrated after centrifugation at the bottom of the tube that you will be examining during this microscopic exam. You will be using both low power and high power to examine this kind of thing, so be aware of that. So we're going to start looking at common cells that you would see in the sediment, because there are quite a few that are relatively common, and chances are we'll come across a few in our own exams in lab. And we're going to start with something you're probably very familiar with, which is red blood cells, right? It's not uncommon to see red blood cells in the urine. Having 0 to 2 per high power field per viewing area is normal, and that's okay. What you're going to look for to identify a red blood cell is obviously the shape is relatively circular, and oftentimes they're going to look a little bit pink. And not always, sometimes they look clear, as you can see in the image up here, but they will give off a little bit of a pinkish hue because they are red blood cells. They have a lot of hemoglobin in them. What you might notice, depending on the urine sample, is that the shape of the red blood cell might change. For example, if you look up here, these little red blood cells that look shrunken are indeed shrunken, and we call them crenated. And that is because the urine that they're in is very concentrated. It's very hypertonic, so the water has left them and kind of caused them to shrivel up. Not uncommon to see them. Make sure you know that when you see something like this, it's a red blood cell. On the other hand, we don't have any images of them here, but you might see one that looks very faint. It looks as though there's not much in it at all. You can just see the outline of the membrane, and that's because the red cell has most likely burst. It was in a very dilute urine specimen, very hypotonic urine specimen, and that has caused it to have water rush into it, burst the cell, and leave just the membrane behind. That's called a ghost cell, but it's still a red blood cell, and it's something you might want to note. One of the things that you can tie to the presence of red blood cells in the urine is potentially kidney damage. You shouldn't really have much in the urine. As I said, 1 to 2 is normal, but that's it. If you have any sort of kidney damage and the kidney is allowing the red blood cells to get into the urine, that is something significant. This is obviously quite a bit of red blood cells here, something to report. There could be trauma to the kidney if you had gotten hit there or fell onto it or something of that nature in a car accident. That's another reason you might see it. There are normal reasons or logical explanations for it too. It doesn't always have to be something terrible. After exercise, sometimes this happens, very strenuous exercise, and obviously women who are menstruating, when they're collecting their specimen, contaminate it, you will see blood in the urine as well, red blood cells. So things to think about as you're identifying them. Something very common, potentially you might see it as you do your own examination. On that same note, we have white blood cells. And different from red blood cells, they're going to be larger. They're not going to have a reddish tint. And one of the things you can kind of see is that they have granules inside of them. They're full of pockets of chemicals. And what they have are little lysosomes with digestive enzymes in it. So if you're lucky, you can see the granules, and that's really going to help you identify this as a white blood cell or leukocyte, another name for it. You can also see the nucleus occasionally. This image isn't super great, but you can sort of tell that there's a nucleus in there. And generally having less than 5 is okay. They're wandering around the urinary tract, keeping things clean, so it's okay to find some of these. If you find more than that, that could be an indicator that you have a urinary tract infection, very common, or also there could be some kidney damage that they're coming in to help to repair. So keep an eye out for white blood cells because they potentially might mean something if in high numbers. Yeast cells are tricky, and that is because they look very similar to red blood cells. One of the nice things is they're generally, and this is a very pixelated image, but generally they are more oval. And one of the key features that you'll see on a yeast cell that's different from a red blood cell is that they oftentimes are budding, and that's basically their form of asexual reproduction. They're going to have a little yeast cell budding off of a larger one, growing off of it. And you're not going to see that in red blood cells most of the time. And that's obviously a sign of a yeast infection. In women, Candida albicans is very common to get. Diabetics also tend to have yeast infections more often or higher yeast concentrations in the urine because their urine is very acidic and yeast likes to live in acidic urine. Also, it tends to have high levels of glucose, which means that the yeast could live there and feed. So it's not uncommon to see it. You shouldn't have any yeast in the urine. Normal is negative, but it's common that that happens. Now, sometimes you will see this sort of hyphae growth. It's more filamentous than just singular here. This is kind of an interesting little setup. It's more of a fungal thing. It might not be just a yeast cell, but it's possible to see something that looks like this and have it be, a fungal infection in the urinary tract. Also, something that you might see would be bacterial cells. They are very, very small, much smaller than a red blood cell, much smaller than a yeast cell. And so what's important is that if you see lots of little specks under your microscope, you may in fact have to go up to your high power objective to see these things. Obviously, they're going to be very numerous in cases where you have a urinary tract infection. And they can have multiple shapes. You might have rods, You might have little spheres, rods are bacilli, spheres are cocci, and sometimes in certain STIs, for example, gonorrhea, you might see them. So it's definitely important to note if you see bacteria. However, due to the collection process for urine, it's not uncommon for contamination to happen. And if you were to collect some urine and maybe let it sit out and not refrigerate it or preserve it correctly, bacterial cells can multiply. Maybe you just had a couple because of urethral contamination. You let it sit for a couple hours and there's hundreds of them. You might in fact think that your patient has a UTI when really it was just a contaminated specimen. So that's why that specimen handling and processing is so important, because we don't want to give back faulty results. Well, they're the right results, but not really for the patient. Often, if you do find that there's a high level of bacteria in the urine, you will follow it up in the lab with a urine culture. Basically, we try and grow what's there because we want to figure out what it is so we can figure out how to treat it. So that's the follow-up procedure if you do suspect a UTI or maybe an STI or something along those lines. Now, other things can be found in the urine, not very commonly, but it's possible. Sperm, you can see the sperm cells here, is something that's potentially in the urine. And it could be male or female, actually. So it doesn't necessarily have to be limited to one gender. Parasites, this is Trichomonas, it's an STI. That is something that is potentially found in the urine. And these are starch granules. Now, this is not something that likely came from the urine specimen. It probably came from maybe the gloves that the tech was wearing while performing the urinalysis. It may have gotten in there. Perhaps there were some on the slide. But the reason I'm showing it to you is because it does sort of resemble red blood cells. And so in that case, or just cells in general, you need to be able to go, it looks like a cell, but something's wrong with it. Something's off. Let me take a closer look. And a lot of times if you just focus up and down on your slide, you'll see that perhaps it's not exactly what you think it is. And so just be aware that there's other things besides those cells that might actually be found in the urine sediment.